get afresh. In our series of histology, we want to talk about muscle tissue. Muscle tissue comes in very many shapes and sizes, and particularly today we want to talk about how it is built. We will not talk about how it works. We will talk about that when we are talking about support and locomotion. So wait for that series as it comes. If it has come, please go and watch it. Skeletal muscles, involuntary muscles, and cardiac muscle are the three types of muscle that we have. So this is skeletal muscle. It is striated, as you can see. It has those stripe-like things. This is cardiac muscle. And then this one here is what we call smooth muscle. You can see this guy here has a bit bigger nuclei. This one has these branches, but these guys here are one cell each. We are going to see why that is so. But first, let's talk about the structure of the muscle structure. So we have, so the first thing is muscle cells. We're going to use the skeletal muscle for the structure. Muscle cells are elongated, thin cells called muscle fibers. So that is down here. Let me get a laser pointer here. These ones here are what we call muscle cells. They are elongated thin cells that are called muscle fibers. Their cytoplasm is called the sarcoplasm. And inside the sarcoplasm, we have a network of membranes called the sarcoplasmic reticulum. Just like we have in a normal cell, we have the endoplasmic reticulum, the smooth reticulum. Here we have the sarcoplasmic reticulum, and it's very vital when it comes to muscle contraction. We are going to see why. And then they are bound by a membrane called the sarcolemma. So instead of having a plasma membrane, their membrane is called a sarcolemma. They possess mitochondria called sarcosomes. So the wording is a bit different when it comes to muscle tissue. Instead of having a normal endoplasmic reticulum, we have a sarcoplasmic reticulum. Instead of having a normal membrane, we have a sarcolemma. Instead of having a normal cytoplasm, we have a sarcoplasm. And then instead of having mitochondria, instead of calling them mitochondria, we call them sarcosomes. Now, striated or skeletal muscle have stripes, and something that looks like this. Okay, and these guys are found attached to the skeleton via a tendon. So I know students always forget it, but tendon connects bone and muscle. Ligament connects bone and bone. Never forget that. And it is attached to a bone by a tendon, which tendon then connects to the outer part called the periosteum. And then the periosteum, we have the membrane. Outward here, we have the outward membrane called an epimysium. Then we have the inner membrane called the perimysium, and then the innermost membrane that surrounds the micro muscle fiber called an endomysium. So an epimysium surrounds a band of fascicles, and then an endomysium, the perimysium surrounds surrounds a band of endomysiums, which are then surrounding muscle fibers. It takes some time to get used to, but you can keep rewatching the video. You can take back and rewatch the video again. So this is how it looks like. I want to go through this very fast. So we have the perimysium inside here, and then inside the perimysium we have the endomysium, which is a smaller membrane, and then under the endomysium we have the muscle fibers. So we have epimysium surrounding the very many fascicles. Each fascicle is surrounded by a membrane called the perimysium. So it wraps the fascicle, and then the fascicle has bundles of muscle fibers surrounded by a membrane called the endomysium. So this is what wraps between individual muscle fibers. It's called the endomysium. So this muscular tissue is attached to the head, the trunk, the limbs. It has muscle fibers, like we have said, that are connected by a series of membranes as they bundle up. And it is important for them to bundle up like that, because for them to bundle up like that, that's how we are going to see how effective it becomes when it comes to contraction. Let's talk about the histology of striated muscle. Now, striated muscle is composed of very many long muscle fibers. That's the first thing we need to know. Composed of very many long muscle fibers. Here, this is the muscle fiber. This is the muscle fiber. We have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. This is an artistic impression of how an endomysium would look like. And then this membrane that surrounds them here has a general name, and that general name is a sarcolemma. Let's remove this laser pointer here. Okay, this general name is called the sarcolemma, and then inside we have a fluid, a cytoplasm called the sarcoplasm, and then the sarcoplasm is bathing myofibrils. Now, these lines here are what we call striations. They look like stripes. Okay, now we are going to see how each of those myofibrils work. And the myofibrils are able to work because they have filaments called actin and myosin. Let's dive in a bit deeper. So each myofibril is divided into light and dark bands. Okay, so we have the light and we have the dark bands. If we go back a little bit, you can see that some parts are darker, other parts are going to be lighter. So it's divided into the light and the dark bands. The dark bands has a light region called the H zone. Okay, so this, even the dark band, so this is where you see these thick red lines, is the dark band, but in between it we have a light region called the H zone, and then through the light zones we have a Z line. So this is the Z line here, like this. This is the Z line. In between here we have the H zone, and then we also have the line that crosses through them called the M line. So the zone is called the H zone, the line is called an M line. When we come to the light zone, the line is called a Z line. So the dark bands constitute what we call A, and the light bands constitute what we call I. The region between two Z lines, from this Z line to this Z line, we call that region a sarcomere. And then these lines that you're seeing through, the thin lines and the thick lines, are alternating thin actin and thick myosin filaments. I repeat, the thin lines are for actin, and then the thick lines are the myosin filaments. Now, why are we calling the dark bands the A zones? That means it comes from the word anisotropic, which means opaque or darker. Okay, so the A is not a letter that we throw up there for just no. It comes from a word called anisotropic, which means opaque or darker. And then I comes from the word isotropic, which means translucent. Not isotopic. I know some chemistry students saying, oh, I have heard of isotopic. No, I'm saying isotropic, which represents the word translucent. So this is a deeper understanding of the myofibril. Let's dive in a little bit more. So actin and myosin, like we have said, actin are the thin filaments, these straight lines. Okay, so this is actin, the thin lines, and then the thick lines, these blue big lines, are the myosin filaments. In between them we have the M line, and then we have the H zone. If we pull out one myosin molecule like this, you're going to see that it has a myosin head, and of course here they are going to be attached to titin. That's something we won't talk about it now, but basically we are saying the thick myosin are fixed and they are confined to the dark bands. For them, they stay in one position. However, the thin actin protein filaments have the ability to extend in between the thick myosin filaments. Like I said, I'm not going to talk about all of it now because some of it is going to be left for the business of support and locomotion. Now, between actin, we have troponin and we have tropomyosin. Okay, we have troponin, we have myosin, and we have troponin and tropomyosin. So tropomyosin is a complex that keeps on blocking the sites where actin and myosin can bond from. Now, when it comes to contraction, we are going to attach calcium. Oh, I'm over explaining. Anyway, we are going to attach calcium to that complex, and when calcium attaches to the tropomyosin, the troponin tropomyosin complex, it is going to change conformation such that actin is going to be exposed. Now, because actin is exposed, the myosin heads are going to bind with actin temporarily. There is a lot of ATP that is going to be involved there. However, this type of relationship is where when someone has ATP, it bonds. Without ATP, it cannot bond. Now, when the bonding is done, the myosin head is going to shift, and then we will be talking about that in a later video. But basically, troponin and tropomyosin, this thread-like thing that surrounds these globular molecules, which are actin, are responsible for blocking the binding sites of the myosin head with actin. Now, during contraction, they are going to be shifted away to expose that binding site. And when that binding site is exposed, then the myosin head attaches on the actin and pulls it inwards. So what will be observed is the sarcomere is going to become shorter. And we say the sarcomere is this space from one Z line to the next Z line. In between, we have an H zone, we have the thick, the dark zone, the A bands, we have the I bands, we have the H zone, and then we have two Z lines. In between, we have H zone. That space in between is going to get smaller because the myosin heads are pulling the actin filaments inwards. Anyway, we will get back to that when we're talking about support and locomotion. Let's do a little bit more and then finish. Each muscle fiber has a membrane system called the sarcoplasmic reticulum. This one here. Now, inside this, we are talking about the myofibrils themselves, which are surrounded by an endomysium. So we have the endomysium is surrounded by sarcolemma. So we have this the sarcoplasmic reticulum is very important because it stores calcium and it surrounds each myofibril. Remember, we have said calcium is very important in the contraction of the muscle. So we are saying the sarcoplasmic reticulum has a system of transverse tubules, these ones here, which are responsible for transporting the calcium once it is released from the sarcoplasmic reticulum. It runs into the muscle fiber along the Z lines. So the T tubules run along the Z lines. Let's add one. Let's go deep one more time. This T system has calcium ions which help ATP to break down. After the muscle contracts, the calcium ions are removed and they are going to be stored back into the sarcoplasmic reticulum. Now, the muscle itself, the sarcolemma, which is the membrane of the muscle, is going to synapse with the motor neuron to form what we call the neuromuscular junction. And we will talk about the neuromuscular junction in our series on coordination, so feel free to watch it there. What are some of the adaptations that make muscles able to do what they do? Number one, they have elongated fibers that provide contractile length. Their fibers can increase and decrease in the length. And then they have parallel fibers which allow individual control. Like we have seen, the myofibrils, they are together, they are in the same cell, but they have like that degree of independency. And then they are tapering and interwoven at the ends to provide strength. Like you saw the tendon, the periosteum bends into it, and then the tendon connects to the bone. And then they have a very large number of mitochondria to be able to provide the ATP that is required. Now, I want to remind you, anytime they ask you for an adaptation, you must provide a description, and at the same time, you must provide an explanation. The actin and the myosin filaments fit into each other, which provides a relationship that allows contraction of the sarcomere. And then they have a rich blood supply to supply oxygen, and then they have myoglobin to store oxygen. And then they have the ability to respire anaerobically for short times to allow muscle contraction even in the absence of oxygen. Like if you're doing a heavy exercise, you can rely on your muscles to take you a bit longer even when you don't have oxygen to supply the process of aerobic respiration. Now, myoglobin is capable of storing oxygen because it has a very high affinity for oxygen, and we will be talking about this in our series on respiration. I know from its very many series. Hopefully, by the time you're watching this, I have uploaded most of them. Let's talk about the types of muscles and get out of here. The visceral muscles, which we call the involuntary muscles or the smooth muscle fibers, they are spindle shaped. This is what I mean by spindle shaped. They look like that. Now, when it is relaxed, it looks neat, it looks spindle shaped, but when it contracts, it takes on a kind of like a squished up shape. They have one nucleus. They are elongated. They are elongated from the ends. They have no sarcolemma. They are fine myofibrils arranged longitudinally inside that cell. They have no striations, so they have no actin and myosin. They are found in areas that don't require vigorous muscle contractions. We have the sphincter muscles, we have the pyloric muscles, the cardiac sphincters, the anal sphincters. Everywhere you have those muscles that close openings, we are going to have their involuntary muscles. And then they have very large mitochondria. Let's talk about them a little bit more. The smooth muscle activity, number one, is involuntary. It's not controlled by you. It's not something you control yourself. It is something that is controlled by the brain, by the autonomic nervous system. There are two nerves that are capable of innervating it. We have the sympathetic and the parasympathetic nervous system. And then they are capable of undergoing prolonged, slow, sustained rhythmical contractions like peristalsis. And they fatigue very slowly. They are found in the intestines, they are found in genitals, in the urinary bladder, we have the respiratory systems, and then they are also found in blood vessels. Let's finish with cardiac muscle. Cardiac muscle is what we call the heart muscle, and it is a network of interconnected cells called cardiac muscle fibers. These muscle fibers are short, cylindrical, and they are branched. Usually, they have one large mitochondria for every one or two nuclei, and then they are connected by what we call intercalated disc, and we are going to look at them. And then for them, they have actin and myosin, but their actin and myosin gives what we call faint striations. So their striations are not as strong as the striations we saw in skeletal muscle. And then their muscle fibers cross and connect. Their muscle fibers cross and connect to form a complex net. So cardiac muscle has a well-developed T system. However, they have a poorly developed endoplasmic reticulum. But hey, it is the reason we are capable of doing the work we do, the heart muscle. Let's talk about it a little bit more. So the cardiac muscle is myogenic. What that means is that it controls its own heartbeat. It is responsible for how it is going to contract. The rate of contractions can be influenced by the ANS, which we call the autonomic nervous system. In other words, the vagus nerve can either slow down, and then your heart rate can be controlled by your brain. However, the heart being myogenic means it can continue beating even without the command of the brain. Okay, and then the intercalated discs ensure rapid and uniform spread of excitation. When we talk about how the heart contracts, you're going to say that it has to contract with a delay. The atrium has to contract faster and in a lower direction, while the ventricles have to contract a little bit after the atrium contract, and they contract upwards. This is possible because of the intercalated discs. They control the direction, the spread of the excitation, and they make sure the spread of the excitation is actually rapid. I always tell people, once your cardiac muscle gets tired, you die. So they never fatigue. They have rhythmic, rapid contractions, and then they relax. And the beauty of it is the cardiac muscle takes long to fatigue. However, it takes a lot of ATP. That's one of the problems that the cardiac muscle has. It takes a lot of ATP. I have a few questions for people who are doers. Write on the adaptations of the cardiac muscle to its functions, compare and contrast the voluntary, involuntary, and the cardiac muscles. Thank you for watching. I know it has been a long video. Thank you for watching. Please feel free to like and subscribe, and have a good evening. Have a good day.